Hello, my name is Mickey Herrera. I'm a current student at UTRGV Edinburgh, and I'll be presenting on uncovering the mechanisms of glioblastoma relapse prevention with niclosamide and camptothecin treatment. Glioblastoma multiforme, or GBM, is an aggressive type of brain cancer. GBM grows at a rapid rate and is resistant against radiation and chemotherapy. Reoccurrence is common with surgery. Because of this, prognosis for this type of cancer is poor. Our study focuses on the use of niclosamide, also abbreviated as N or NICL, and camptothecin, also abbreviated as CPT, against GBM cells. Niclosamide is an anti-helmintic drug that disrupts cancerous cells' metabolism and signaling. CPT is a topoisomerase inhibitor. It inhibits enzymes essential for DNA replication and transcription. We hypothesize that the use of niclosamide and CPT on glioblastoma cells will produce sustained anti-proliferative effects due to potentiating disruption of oncogenic signaling. We use different type of assays in order to test our hypothesis. The cells that we used were U87 cells, which are human line cells derived from the glioblastoma cells. We cultured them in DMEM medium with 10% FBS and 1 part penicillin and streptococcus antibiotic. Cells were left to grow on plates for 24 hours or until they were at 80% confluence. They grow fast, so 24 hours normally did the trick. They were then treated with niclosamide, CBT, and a combination of both drugs. After 24 hours, the drugs were taken out, rinsed, and once again put in with just regular medium. The cells were observed using light microscopy right after treatment, 24 hours after treatment, and 48 hours after treatment to see their recovery. Along with light microscopy, MTT and tunnel assays were performed. Once again, 24 hours and 48 hours after recovery. MTT assay is a test used to measure cell viability by viewing the number of metabolic active cells. A tunnel assay that was also used is an assay technique that's used to quantify apoptosis by staining fragmented DNA strands. For the tunnel assay, we did have to use fluorescent microscopy. Along with the assays, we did also place a caspase-3 marker following treatment and recorded caspase signaling proportions. Caspase-3 is a marker for apoptosis. It's a type of enzyme that's released in programmed cell death. In our results, we can see first our observations using light microscopy in the panel towards, or the first image towards the left, or we can see that we have control, niclosamide, CPT, and the combination in that order going to the right. We can obviously see that the control is a lot denser than the combined treatments, and the density does lessen as it goes down the line. So niclosamide, we do see that it does lose some density compared to control, and that continues with CPT and with the combination. For the MTT assay results, we made some histograms. In these histograms, we can see that the metabolic activity recorded in the U87 cells following 24-hour treatments. The MTT assay conducted at 24 hours and 48 hours post removal of the treatment from the cells, including wash and medium changes. We separated the histogram, and they indicate that statistically significant results. Same histogram was used, or the same type of histogram was used for the caspase-3 recordings. For these recordings, we used a cell counter following 24-hour treatments of the U87 cells and a 48-hour recovery period. And then at the very bottom, we have our tunnel assay results. So we did have to use fluorescent microscopy to see these results. So once again, towards the left is control, followed by niclosamide, CPT, and the combination. Um, and these images, the blue, the little blue dots will indicate um, live cells. So this is or it indicates the nucleus. And then green dots will indicate cell death, which is just the DNA fragments. So in the control, we can only see um, blue or blue cells. The niclosamide, we can also mostly see blue. Once we get to CPT, we start seeing a couple green dots, which indicate DNA fragments, so cell death, towards the top left corner. And then for the combination of the treatments, we can see that it's mostly green dots, and there's almost no live cells to be found. So the combination of niclosamide and camptothecin exhibits potential and sustains anti-proliferative effects on GBM cells mediated through enhanced apoptosis. The niclosamide or camptothecin treatments alone demonstrated variable effects on caspase-3 activation and metabolic activity. A significant decrease in cellular density and cellular metabolic activity, paralleled by increases in caspase-3 activity and apoptotic signaling was noted in GBM cells, giving the combined treatment. These findings highlight that the potential of a combination therapy with niclosamide and camptothecin as a promising treatment strategy for GBM. The current study has shown positive results supporting the proposed hypothesis. The methods we used were tunnel assay, automatic cell counters. However, both these methods are extremely time consuming. Additional trials will be needed to run more data collection. In addition, we would also like to do Western blots in the future in order to identify apoptotic signals and identify the signaling pathways in which the the drugs are the tested drug combination produces sustained anti-proliferative effect on GBM cell lines. I would like to thank you for your time and attention, and I would also like to give a special acknowledgement to UTRGV School of Medicine, UTRGV Biomedical Research Institute, and UTRGV College of Science, as well as everybody who has helped with this project.